Now let us understand counter current mechanism. What is counter current mechanism? Now the urine gets filtered in the proximal convoluted tubule. This is proximal convoluted tubule, this is distal convoluted tubule, and this is the collecting tubule. So now it is collected in the proximal convoluted tubule. The osmolarity of this fluid filtrate is same as the blood and the interstitial fluid, the fluid which is present in the cortex. It is isotonic with this fluid, so it is 300 milliosmol. Now, as the filtrate goes on, its concentration goes on increasing in the descending limb of the Hillis loop. Why the concentration goes on increasing? Because this is permeable, the descending limb is permeable to water. As the water goes out, the solvent is gone out, the solute is what? Urea and salt, that has increased, NaCl is increased, urea is increased. Water has gone out because this is permeable only to water. So when little water goes out, it becomes 400. More water goes out, it becomes 600. More water goes out, it is 800. More water goes out, it becomes 1000. More water goes 1200. Here, as this limb gets over, finishes, enters the U tube, it becomes 1200. Now, permeability of water is present only up till this region. Now, as this tube goes into the ascending limb, the water will not enter or will be given out. It is impermeable to water. Here in the ascending limb, it is only permeable to NaCl. NaCl will be given out, and now reverse process will take place. Concentration had taken place up till this region. Now the solvent is not going out, solute is going out, the NaCl is going out. So now it will become dilute. Solvent will remain as it is, solute goes out, so it becomes dilute. As it goes up, now 1200 to 900 to 600 to 400 to 300 to 200. Now it has reached in the distal convoluted tubule. It will be 200 milliosmol. Now here it will enter into the collecting tubule. In collecting tubule also water will be given out. Absorption of water will take place. What we require is concentrated urine with more of urea. So water has to be absorbed. Our body requires water. Our body requires NaCl. They are there in the body itself. What is going out is urea. It will go out from the collecting tubule. Now remember, some urea is given out in the interstitial fluid of the medullary region to maintain the osmolarity of this fluid. Some little urea is given out which will be absorbed by the ascending limb. But to maintain the osmolarity, osmoregulation of the interstitial medullary fluid, the fluid which is present in the in between, which is present in the medulla, the concentration has to be maintained in this fluid with the help of urea. Now, why it is known as counter current? Because there is a current which is formed in this region, in the descending limb, and there is a current formed in the ascending limb which are exactly opposite. Since they are exactly opposite, they are known as counter current. So going down, the concentration gradient going up here and then going down, they are exactly opposite. The concentration gradient that is increasing and then becoming less and less, they are exactly opposite. Therefore, they are known as the counter current. Now this counter current helps in concentration of urine. We are interested in concentrating the urine here so that water is absorbed, NaCl absorbed, which is required. So counter current helps in concentration of urine. Now, hugging close to this Henle's loop is vasa recta. Vasa recta is network of capillaries. Capillaries will have blood in it. Here, water, urea, NaCl is there in this Henle's loop. But here in capillary, blood will be there. That is tightly hugging, very close, running parallel to the Henle's loop. The counter current will run in this vasa recta also. Now, this ascending limb is giving out NaCl. This NaCl will be, now this is ascending. So, it will go into descending. Opposite. This is ascending, which is giving NaCl, goes to the descending. Ascending will give to the descending. Now this descending will give to this ascending of vasa recta. So from here it will go here and from here it will go here. So this is ascending limb will go to descending vasa recta. Descending vasa recta will go inside here. Then it will give to the ascending vasa recta. So here also exactly opposite gradient have been formed. The concentration is exactly opposite of ascending and the descending and the ascending limb. So there is a counter current which is formed in vasa recta also. So you have Henle's loop, surrounding you have vasa recta. The counter current is taking place in both descending, ascending. Goes on in concentration, goes on increasing, then decreasing. Why? Here because water is given out. Here why? Because NaCl is given out. And this NaCl is taken by the descending vasa recta. And from descending vasa recta, NaCl comes to ascending vasa recta. Here blood is present, here water and urea and NaCl is present. So here blood will take in NaCl, give to this ascending limb, and then that will balance the concentration of the intermedullary fluid, the fluid which is present in the medullary region, interstitial space. The space has got liquid. That concentration has to be maintained, which is very important, so that the function of this collecting tubule helps in concentrating the urine. So now this counter current helps in absorption of water. Absorption of water takes place in the collecting tubule and the urine, which is concentrated 4 times due to this counter current mechanism.